Okay, once we start getting the microarray data, you get your CDF file, you get your cell file. The first thing we need to do is to do normalization. A very simple reason we need to normalize is situations like this. You look at one array, let's just say the same sample, you hybridize on the array on different arrays on different days, or even if you hybridize to the same array, but you run this array on different scanners, it might come back showing, for example, this whole array is darker and that whole array is lighter, even though it might be the identical RNA, which means, maybe you labeled your RNA a little better, or that scanner setting is set slightly different. It doesn't mean that the RNA in the first sample are all higher than the RNA from the second sample. So you have to kind of normalize the data before you can do comparisons. So for normalization, imagine the case where you have for normalization, we try to preserve the biological variation in the actual expression of different genes, but try to minimize any technical or experimental variation, so the different experiments can be compared. One important assumption for normalization is we have to assume that majority or most of the genes or the probes between the two samples or between the two conditions are the same. You can't really compare something where most of the things are different. Then you don't have a baseline, right? So we have to make some basic assumptions, which is probably true. If you look at individual, like human tissues, even though our brain cells and our muscle cells or our blood cells, there are a lot of differences, but still most of the genes are expressed at similar levels. So we have to at least make that assumption in order to do normalization. And this is a very important step. If you don't do the normalization right, it can have even larger effect on analysis than any of the downstream things that you do. And so make sure we do this correctly, okay? So the very, very simple approach for normalization are cases like this. When we say array 1 or array 2, they are the same array type. We never try to compare or normalize arrays, or people tried, it never worked very well. Say the same sample hybridized to a U95 array and also hybridized to U133 array. Don't try to do that comparison, it's just too crazy. Okay, make sure it's the same array type, but say you have two samples. This is like sample 1, this is sample 2, and you look at all the probes on the array, and if our assumption is right, then most of them should be the same, right? This is the ideal case. We want them to be like this. They have roughly the same median, and they have the rough same scale. And so a simple normalization like this, if you see something like this, you will know that, okay, we need something called a median scaling. And just to make sure that the median is comparable and the scale is like really, this line is really on the diagonal. And you do something like this, very simple. However, sometimes you might see cases like this. It's not linear. There might be some signal plateauing at certain time, at certain level, the signal is really bright. The yellow has already reached a plateau. You can't get better signal on this one sample, but the other sample keep going higher. So how do we normalize that? There is an algorithm called LOS. You can weight or not weight, either way, it's kind of similar. The idea is we are trying to estimate kind of a curve along the mode or most of the points on this graph, but it's difficult to estimate the whole curve. Instead, we piecewise estimate a linear. So you look at the spot in here, you estimate a linear scale, like a slope. And then you look at the next region and you estimate a linear scale. And you estimate, piece by piece, and then you connect the linear, and then you'll get a kind of a curve. And so this way, you can imagine the original curve might look like this. It's not a line, but after you can just run the R function for LOS, it will normalize, you will get something like this right side. Okay, then you know roughly normalized it correctly. The difficulty here is when you try to normalize, you're always using one sample as your reference and the other sample as the weight. There's no guarantee that the reference you are using is really good, right? Which one do you want to use as a reference? It could be tricky, especially when you have a situation where you have, let's just say the very simplest case. You have a microarray, you have the cells growing in Petri dish. You have 3 replicates of the cell just growing happily on their own. And then another condition is you treat the cell with some drug. And you have 3 replicates of this. You have 6 samples there. Which one do you want to use as a reference? It's a little harder to do, right? So what ended up being a very popular approach is called quantile normalization. And this is developed by Terry Speed's group at UC Berkeley. And this is a biostatistics group, which is great. You can see actually throughout the course, we will learn a lot of algorithms. Some of them are developed by statisticians or biostatisticians, and some of the algorithms are developed by computer scientists, and they each contribute different aspects. Anytime I would say most of the time, when you're dealing with numbers and counts and normalization, the statisticians do a better job. And anytime you deal with like just sequence comparison alignment, you read this base being different, usually that's a computer scientist, you can see. So this quantile normalization was developed by statisticians in here. So the idea is, you can imagine if we have, in this case, 6 samples, right? 3 controls and 3 treatment, 6 samples. Each row here is a probe value, and on this axis array, you might have millions of rows, and each column is a sample or an experiment. The different values, in this case, red is the highest value, purple is the lowest value. In order to do quantile normalization, we create another column, which is the mean column. We'll see how this is done, okay? So you can imagine, let's imagine this is Ivy League schools. Students get different GPA, right? And in order to really make sure that you can compare, we're assuming the Ivy League students are all roughly the same, right? And we know there's Harvard, there's great inflation. And we want to compare a student from Harvard with a student from Princeton, okay? So what we do is we take the student that has the highest GPA from Harvard and also the highest GPA student from Princeton and from Dartmouth, from Yale, Brown, and Cornell, whatever, Columbia. And you calculate the average of the best student GPA, and you put this on this mean row. And then you take the second best students in each school, you average their GPA, and you put this on the second. And you do this across all, assuming they have all the same number of students, okay? This way, this mean would be the average GPA of the best student, the second best student, and the average GPA of the worst student from every school, okay? Then we can use this column to replace the original data. Now the best student, instead of its original GPA, will get this mean value. The second student will get the second average. The third student will get the third average. And we use the original column to replace all of these other values. The order is different, right? So you can see this is one probe. This probe might be the best in the second sample, but it's not the best in the third or the fourth samples, right? So it will be a different value, but basically after normalization, the highest scoring probe in every sample will have identical value, which is this mean. The second best value in all of the arrays will have the identical value, which is the second in this column. Okay, so this way, the assumption is hold. We assume on this array, majority of the things are comparable or are the same. And we'll make sure that every array have the identical distribution of the probe values. And the nice thing there is, initially when you only have 6 samples or 10 samples, if you add a new sample, let's just say you already have 10 samples, you add an 11 sample, you'll need to recalculate this mean because now the average from the 11 numbers is different from the previous 10 numbers. However, supposedly you already have 100 arrays from the same array type, 100 different conditions. You created this mean column, and now you have the 101 array. By that time, you don't need to create this mean anymore. You just say, okay, rank this last array, replace the top one probe, second probe, third probe with the mean that you already got from the previous 100 arrays, you'll probably get a pretty decent number. So this way, no array is considered a reference sample. You create this reference sample and you use it to replace all the original data. Okay, so that's quantile normalization. Let's see how well it works. This is a dilution series where initially you have one RNA sample. You dilute them by half, just like 1 to 2, and then you dilute by half and you dilute by half. And each of the sample are then processed and sent to 5 different array scanners, different machines to process. And because of the processing, because of the scanners are different, at the end, this is what you get. This is the highest concentration sample, the original sample. When you put it in 5 process and scanners, this is the original probe distribution. Then this is like half of that concentration. This is like a quarter of the concentration, and 1/16, and then so on. Okay, so you can see a little bit from here. That first machine, from this one, it's always a little higher. I think the machine is tuned that the signal is stronger. And the second machine, it's always a bit lower, right? But then once you do quantile normalization, so I want to ask, do you want to quantile normalize all of the samples together, or you only quantile normalize the samples within the same color? You want to quantile normalize samples for the same color because the assumption of normalization is that all the samples have the same distribution. The yellow and the green, you know their total RNA amount is different, so you don't want to normalize that, right? But after quantile normalization, not surprisingly, the 5 green samples are now identical because they're all being replaced by your original, or by your average column there, right? So they are kind of comparable now. And in order to see whether we have really normalized it well, we use a plot called MA plot. You will see MA plot a lot when you look at gene expression. Imagine the original study, if we just look at a pair of samples. So in here, each of the condition, we have 5 samples, but let's just compare a pair of samples. We call them R and G. So because very early days, the one sample is labeled red and the other sample is labeled green. And every dot here is one probe. If you normalize them well, the probes or these 2 samples should have most of the dots on the diagonal, right? You can see from here it's not so great. In order to show this in a slightly different way, the MA plot is like this. The M is the log first value minus the log second value. And this is the difference in some sense, the log difference between the 2 samples, right? And so that's the M. This is basically looking at how different it is. And the A is, you can see here, is the log one sample plus log the other sample divided by 2. This is the geometric mean, right? So this is, I can now write directly in here, is this, red and green, right? That's what the A is. It roughly calculates the average expression of this gene between the 2 samples, okay? Oh, sorry. I need to clear my drawings. Do this. Okay. Oops. Yeah, so Now, basically, you want to see at a different level of gene expression, are there differences of every gene across different range of concentration. So if the data is normalized well, what you would expect is on this diagonal plot on the left, most of the dots should be on the diagonal. You might see this is okay, but if you run this on an MA plot, you can see there's definitely a bias. What we expect to see is across all range of concentrations of different genes expressed at different levels, the two samples are mostly comparable. So the dots should lie around the 0 on the on the y-axis. Okay, and so let's go to the original dilution series. We need to let's just look at the 5 arrays in the 5 different scanners. The original data, because there are 5, we have to do pairwise comparison of every pair. So MI plot, you compare every pair, and the red curve is the low S, which is kind of, remember, piecewise linear. You can see that kind of the average trend. You expect most of the dots to lie on this blue line, but based on this, you can see the original data definitely need normalization. Most of the lines do not lie on that 0, right? So that's why we need to normalize. So in here, the M again is the difference, and the A is the geometric mean, right? And you can see the data clearly need a normalization. But interestingly, even just after quantile normalization, most of the dots now really lie on the 0. It means, so when you look at the raw data, if the quantile normalization look like this, you think you have done a good job with normalization. Okay. Yeah, so questions? Yes.
When we normalize across samples, do we need to have the same number of
Yes. With Affymetrics, so early the Affymetrics and later the Affymetrics have different number of probes on the same array. Their location on the probes also different. And depending on the location, their signals change a lot. People have tried to normalize against those different array types. We would not recommend that. Just the same type of array, different samples, you can do this normalization. Okay. Other questions?